Hi, kids. All right, we're going to tackle a really tough topic here that will start off with something familiar from first year bio, but then go a little deeper into things. No pun intended, since we're talking about water here. But anyway, osmosis. We know osmosis is the diffusion of water along its concentration gradient. So let's write that down. Diffusion of water along its concentration gradient. And it's important also then to add to that definition across a selectively permeable membrane. Okay, so got to take into consideration the membrane and what we've learned before with active and passive transport. Osmosis is a type of facilitated transport, facilitated diffusion, because that water has to go across the proteins in the membrane. So across that selectively permeable membrane, it's definitely the proteins that are selecting the permeability of that water, because the water is not going to go across those phospholipids. That's oily. And oil and water just don't mix, okay? Then the concentration gradient, okay? We know things flow from a high concentration to a low concentration, okay? We know that from first year bio. We know that just from like experience. So what I have set up here is I have 3 beakers. And in each beaker, I have a model of a cell that is 85% water and 15% solutes, okay? It's an aqueous solution. Water is the solvent. Solutes, let's say it was sugar this time, or it could be salt. Whatever that solute is, that will come into play though later on. Okay, and something that's very important to remember here with osmosis, the principal mechanism of osmosis is the concentration. So let's write that down. Principal mechanism is concentration. Now, in first year bio, we just say percents, okay? But we learned something else in the beginning of the year. We learned molarity, okay? And we will see how molarity comes into play in a little bit. But let's just look at the percents right now. Now, I'm going to say that I have the same type of cell, 85% water, 15% solutes in each of these beakers. But in the one, I'm going to put an isotonic solution, isotonic. In the next, I'm going to put a hypertonic solution. And in the last one, I'm going to put a hypotonic solution. Okay. Iso is a prefix that means same or equal. Hyper is a prefix that definitely means like more. Hypo is less, okay? And when you're talking about the tonic here are the solutes, okay? Isotonic, the solutes in that solution. We're talking about these solutions like right in the beaker, not what's inside the cell. We're talking about the solutions in there. We're saying we're going to put this cell in an isotonic solution. So that means that the solutes in the solution are going to have to be equal to, so they will be 15% solutes in this solution, and therefore the water is also equal to what's inside the cell, 85% water. And in this situation, we're definitely going to have water is going to flow equally in both directions. So water flows equally in both directions. Because we say that this is at equilibrium, at equilibrium. Okay, so that was pretty easy to figure out. All right, let's take the next cell. And we're just saying it's a hypothetical cell. We're not saying even if it's a plant cell or an animal cell yet. But in this situation, we have a hypertonic solution. So that means the tonic, okay, has to be more. And the tonic is always the solutes, Okay, so here again, 15%, but we have to have a higher thing. So if we were making a hypertonic solution, we'd have to have more than 15% solutes. So let's say it's 25%. That's definitely something more. 25% solutes out here. So what would that make the water? If this is 25%, it's got to equal 100. That would make 75% water. Now, notice here we said water flows equally in both directions because we're talking about osmosis here. Okay, we're not talking about the solids, and that's why I just call them solids. I didn't call it glucose. I didn't call it salt. It could be anything. We're talking about which way is the water going to flow. And here, the water were both equal, okay, just like the tonics were equal. But here, this time, because the solids are greater outside the cell, it makes the water less outside the cell. So if we're looking at which way is the water going to flow, well, 85% inside, 75% on the outside, it's definitely going to flow. And let me get like a different color here to like just highlight the different colors. Okay, it's definitely going to flow out of the cell, out of the cell. So water flows out here. Water flows out. And what will that do to the cell? Well, it'll shrink the cell. Cell shrinks. Okay, it shrivels up. Okay. This, if you remember from first year biology, this is called plasmolysis. Plasmolysis. Or we could actually say, that if this was a plant, we don't always say plasmolysis for a plant. You can. We could say, or it has low turgor pressure. Turgor pressure. Okay? All right. And finally, our last one, hypotonic. Hypo is less with the tonics. So the solute in this case was 15%. So it's got to be something less. Let's make it 5% solutes out here. So that would make 95% water on the outside, okay? We could have even put a distilled solution there because then the solids would have been 0, okay? But anyway, we want to know osmosis. Which way is the water going to flow? 95 is greater than 85, so we see the water is going to flow into the cell, okay? I used to always say, remember, hypertonic, shrivels up the cell. Hypotonic makes the cell swell up, okay? So in this case, water flows in, and the cell swells. And if it was an animal cell, it would burst because it doesn't have a cell wall to protect it. But if it's a plant cell, it would just have high turgor pressure. So cell swells, okay, and could burst, which is called cytolysis, or just have high turgor pressure. High turgor pressure, okay? So that's what we did like way back in first year bio, okay? But we're going to add like a little twist to it. We're going to say, well, what if we were dealing with molarities, okay? Let's see. Remember, and let's do this, remember that concentrations can be expressed in molarity. Remember, molarity. Okay, molarity equaled the moles of the solute, right, per liter of solution. So you're looking at how much solute per that. So the higher the molarity, the more it is concentrated, okay? So let's say we have that same cell, and we're going to compare it differently. Let's say we have that same cell, but this time we're going to give it a molarity of 0.5, capital M, okay? And we'll say this is a sucrose solution. Okay, and if it has 0.5, well, what would make it isotonic? What would make an isotonic solution. If we put this cell in an isotonic solution, that isotonic solution would also have to be 0.5 molarity, okay, because they have to be equal, okay? But what about if it was let's do hyper first, hypertonic, so let's say in the same direction. Well, then that would have to be anything higher than 0.5, higher than 0.5 molarity, okay? And hypotonic would be lower. Hypotonic would be lower than 0.5 molarity, okay? So up here in these pictures, I could have wrote 0.5 molarity here. It would be 0.5 out here. Could write 0.5 here, and I would have had to say something higher, like maybe 1.0 molarity. And maybe out here, since it would have to be lower, I could have said 0.2 of a molarity. Okay, it'd have to be lower. So that's a way that we can look at molarity in those situations. Okay, but here's the problem with osmosis. Way back in first year bio, we just looked at the concentrations, but there's so many more things that are going to come into play. So let me turn the page here. I have this second page here, okay? There are still other factors that affect the diffusion of water. Sorry, you couldn't see that at first. There are still other factors that affect the water movement, okay, the diffusion of water. So can you think of some of those factors? What else would affect how these particles are going to move? Well, first off, the particles themselves. The particles themselves. Are these particles molecules or are they ions? What are they? Molecules or ions? Molecules or ions. Okay, that's very important because if it's a molecule, it stays whole as like one being. But if it's an ion, it breaks into two. It actually doubles the amount of things. Okay, that's particles. Second thing you got to consider, what else is going to affect the movement of particles? Well, if you just think about what measures the motion of particles, that's temperature. So temperature, which is, the average kinetic energy of the particles, okay, temperature is definitely going to have an effect. And then if you remember from your chemistry days, temperature and volume, and one other thing that was really important was pressure, okay, pressure. Okay, so there isn't just, we got to grow up here. It isn't just all about the concentration. There's other things. So what we do in science is we don't just look at concentration anymore. We look at something we call water potential. It has this funky symbol that's like, I think it's a Greek letter called a psi. I don't remember my sorority and fraternity letters days. But anyway, water potential is going to predict which way water will flow. And this mainly occurs in plant cells, but it takes into consideration all these factors. So it predicts which way water flows, mainly in plants, because we care about plants. They have to have that water for photosynthesis. And it takes into consideration these other factors. Takes into consideration all factors, not just concentration. Okay? All right, so this video is going to end, so I'm going to have to stop this, and I'll pick it up right at this, and we'll get into the water potential equation.